AP Biology, second quarter review, part 1. We started the quarter with the cell and the cell theory. The three parts of the cell theory are all organisms are made of cells. The cell is the basic living unit of organization for all organisms. Below the level of a cell, we don't consider that alive. It doesn't meet the characteristics of life. And all cells come from pre-existing cells. Now, this doesn't talk about the first cells on this planet. We'll talk about the hypothesis as to where cells came from in a future class. Activities of life. So these are the things that all the cells can do independent of whether the organism is single-celled or multicellular. All cells can reproduce, grow and develop, use energy, respond to the environment, and maintain a stable internal environment called homeostasis. Below the level of a cell, the molecules that make up the organelles can't do that. How do we study cells? Using microscopes. They've advanced our understanding of how the living world works. We have light microscopes that pass light through a specimen, and they can be used on living cells, so that's useful to see things while they're still alive. And they can see down to about the level of the larger organelles like mitochondria and some of the bacteria. Electron microscopes are more powerful than light microscopes. They can see even smaller. They rely on coating the specimen with gold, which disrupts the function of those cells, so they're no longer living. And they're only used on dead organisms. But they're able to see a lot smaller. And we can see down to the size of some of the larger atoms with electron microscopes. There's two types of electron microscopes. Transmission electron microscope is more powerful. That sees in 2 dimensions. Scanning electron microscope sees in 3 dimensions and is less powerful. But they're both more powerful than light microscopes. How do we separate out the organelles within a cell? The process is called cell fractionation. The first step is basically just blendering up the cells, breaking open the cell membranes. And then the second step is to centrifuge out the cell parts. The more dense organelles settle to the bottom, the lighter organelles settle to the top. You should know that the more dense organelles include things like Mitochondria and Chloroplasts, and the lighter organelles are the ribosomes. Keep in mind these are things that all cells have. So you get questions like that sometimes. If you see a cell with blah, 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 well, if the cell has these 4 things, then it could be any cell. It could be a bacteria, it could be a plant, it could be an animal, it could be a proteus, a fungi. It really doesn't help you at all because all cells have these 4 things in common. All cells have a cell membrane called a plasma membrane made of phospholipids. All cells have a cytosol. Cyto means cell, sol means solution. Basically just a liquidy interior made mainly of water. The cytoplasm is the cytosol, the liquid, plus the organelles floating around in it. Sometimes these words are used interchangeably, but these are the definitions. All cells have chromosomes, which have genes in the form of DNA. Now, remember that eukaryotes have their DNA in a nucleus, a membrane-bound container. But all cells, including bacteria, have DNA. And this is one thing that binds all of us together on this planet as a commonality. And these DNA are highly coiled in the form of chromosomes. And all cells need enzymes to make all the things in their cell, and you're only going to make those protein enzymes with ribosomes. So all cells also have ribosomes. And remember that the bacteria have a smaller ribosome, but everything's going to be making proteins from the ribosomes. Ribosomes are not membrane-bound organelles, so they're not unique just to eukaryotes. These are some helpful Venn diagrams. Pro means before, karyo means nucleus. These things evolved before nucleus. However, you can use this little mnemonic trick to help you remember this. Prokaryotes, pro means no membrane-bound organelles or nucleus. Now remember, the ribosome is membrane or not membrane-bound, so that is something that prokaryotes have. However, look at all those things that eukaryotes have that are membrane-bound: nucleus, endoplasmic reticulum, Golgi, lysosomes, vacuoles, mitochondria. All these are membrane-bound organelles that are not shared with prokaryotes. Prokaryotes, another difference is that they have circular DNA, and basically it's a DNA, double-stranded DNA looped in a big circle. We have eukaryotes that have strands of DNA, and they're more functionally complex as well as a little bit bigger, about 10 to 100 micrometers or microns in diameter, whereas prokaryotes are between 1 and 10 microns. Just to give you a sense of scale, a human hair diameter is about 20 microns. Remember, prokaryotes include bacteria. If you hear the word bacteria, you're talking prokaryotes. If you hear the word prokaryotes, you're talking bacteria. So there's only one type of organism that's prokaryote, and that's the bacteria. The eukaryotic kingdoms include plants, animals, fungi, and protists. Now, even though things like yeast are single-celled organisms, they are eukaryotes, and they have a lot more things going on inside than a single-celled prokaryote, like bacteria. And two types of eukaryotes, animal and plant cells, you should be able to compare and contrast. Notice all the similarities between animals and plant cells. We have a lot in common. In fact, 50% of our genes are the same as things like corn, and a lot of that stuff is what goes on inside the cell. We have the same type of ribosome, nucleus, endoplasmic reticulum, Golgi, vacuoles, mitochondria, cytoskeleton, holding things in place. Remember that animal cells have two things that plant cells don't. Lysosomes, they are sacs of hydrolytic enzymes that break down things. Plant cells have their enzymes just floating around their cytoplasm. Centrioles are attachment points for spindle fibers during mitosis. Animal cells have them. Plant cells have a cell wall to attach their spindle fibers to. Plant cells have a cell wall that we don't have. Don't get cell wall and cell membrane confused. A cell membrane is going to control what goes in and out of the cell, made of phospholipid bilayers. Cell walls, made of cellulose, provide a lot more protection and support for the plant cell, and they are one of the reasons why a plant doesn't fall over. Plant cells also have the energy capturing chloroplast that takes light energy and converts it to chemical potential energy of sugar. And plant cells also have a large central vacuole for storing water and ions inside that we don't have. We have vacuoles, but they're not big like that. All right, cell types of the 6 kingdoms. The prokaryotic kingdoms are bacteria. If you see bacteria in the name, you know you're dealing with a prokaryote. Archaebacteria and eubacteria are both prokaryotic kingdoms. Eukaryotes include animals, plants, protists, and fungi. We'll talk more about classification third quarter, at the end of third quarter. So what limits the cell size? Well, the short answer is small cells have a greater surface area to volume ratio. That's a key phrase. The basic idea here is that if you had this box here and this box here, or a whole bunch of little boxes, they both have the same volume here. They take up about the same amount of space. However, there's, in addition to the outer surfaces, for a whole bunch of little boxes, which would represent cells, they also have the inner surfaces as well to transport things in and out of the quote-unquote cell. So if you take a look at both these pictures, you can kind of visualize which one's going to have more surface area compared to the volume. They have the same volume, but this one here has a lot more surfaces for things to move in and out. So why is it important for cells to have a greater surface area to volume ratio? Well, the answer is moving stuff in and out allows for faster metabolism. You are able to move things like glucose through more surfaces, oxygen in, which supports a higher metabolism, which allows you to do things faster. And if you can imagine the effect on a large organism like a bunny or something, a bunny that can move faster versus a bunny that can move slower, well, what's going to have a better chance of survival? So small cells were selected for nature because they allow for faster exchange of materials. Organelles and internal membranes. Eukaryotic cells have all kinds of extra membranes inside, membrane-bound organelles. They partition the cell into compartments, so that's a general idea of what organelles are all about. They can compartmentalize the functions. Like when we have a lysosome, we can compartmentalize those hydrolytic enzymes to prevent it from digesting the rest of the cell. Same with the mitochondria, building up proton gradients. You can't do that unless you have some kind of compartmentalized space, like the inner membrane space between the inner and outer membrane. All right, so we're going to get into eukaryotic cell organelles now. Here we have a generic animal cell. Animal cells don't actually look like this. You have about 100 different types of animal cells in your body, and this is kind of a composite of a lot of characteristics of many of your cells. Also, they don't kind of stand alone like this unless they're blood cells kind of floating around in your blood. They're attached to other cells as well. So here we have some of the structures we're going to talk about. And then in plant cells, we got some extra stuff going on. We got the green chloroplast, make plants green. We got the cell wall here, little holes in the cell wall called plasmidesmata. Remember that plant cells also have a cell membrane. They have both. There's the large central vacuole that we don't have. All right, so the first organelle we're going to talk about is the nucleus. Nucleus controls the cell, basically. The genes, the DNA is held in the nucleus. And there are some genes located in the mitochondria and chloroplasts that they probably kept from when they were bacteria, and they still have some function. But most of the information, the genetic information, is kept in the nucleus in the form of DNA. What's the nucleus structure? Well, the nuclear envelope is a double membrane, another plasma membrane, a cell membrane basically, but on the inside. They have little pores, little holes, that's what a pore is, to allow large macromolecules to pass through. When we talk about this next quarter, we're going to talk about how messenger RNA leaves the nucleus and attaches to ribosomes out in the cytoplasm, and the other things will enter the nucleus, like the building blocks for DNA and messenger RNA. Nucleus structure, now don't get this word confused with nucleus. Nucleolus is a darkly stained part inside the nucleus. Nucleolus, nucleus, and you can imagine the whole page is like so. The nucleolus is involved with the production of rRNA, ribosomal RNA, and proteins. And these are the two parts that make up a ribosome. So make sure that you know that nucleolus makes up the two parts that make up a ribosome. And there's a large subunit and a small subunit that's made in the nucleolus. That also will, of course, have to pass through that nuclear membrane. Ribosomes, think ribosomes. Ribs have protein on it, and they make proteins. So putting together the primary structure of the protein, remember that the primary structure of any protein is just a peptide bond sequence, a type of covalent bond. Then you have the secondary structure made with alpha helixes and beta pleated sheets. Then you have the tertiary structure, which is a combination of a whole bunch of things, like the hydrophobic and hydrophilic interactions, as well as ionic bonds, hydrogen bonds, and disulfide bridges to give you that final three-dimensional shape. And the shape is really important to a protein. So the ribosome puts together the peptide bonds in the protein. And remember, a lot of proteins are really important. They're enzymes. You can kind of think of a ribosome as a super enzyme, putting together the things that will put together other parts of the cell. There's two types of ribosomes, free ribosomes and bound ribosomes. Now, the free ribosomes are just floating around the cytoplasm. They're not attached to anything. And they're going to be making the proteins, remember ribosomes make proteins, that are going to be functioning within the cytosol, if they're like enzymes and other things that are going to put things together. So if the protein is going to be used by the cell, the protein was made by the free ribosome. Then we have another type of ribosome called the bound ribosome, and they're attached to the rough ER. In fact, that's why it's called rough ER. It looks rough because of all those little ribosomes. And they're going to be making proteins for export out of the cell. For example, your pancreas is going to make lots of digestive enzymes that are sent out of the cell. Mitochondria and chloroplasts, they are both involved with transforming energy. The chloroplast transforms light energy into chemical potential energy, and it's like glucose, a stable form of that energy. And the mitochondria takes that glucose, breaks down that stable glucose, and makes a unstable molecule called ATP. And ATP is unstable as a result of that third phosphate not being held on very tightly, and is able to be transferred very easily to do work. But they're both involved with transforming energy. Light to chemical potential energy, and then for mitochondria, one type of chemical potential energy into a more usable form called ATP. They both have double membranes. They both are semi-independent or autonomous of the rest of the cell. They move, change shape, and divide independent of the rest of the cell. And they have their own ribosomes on the inside, their own circle-shaped DNA, and they make some of their own enzymes. Now, they can't live outside the cell, but we think at one time they did. Mitochondria function as cell respiration, and the main purpose of that mitochondria is making that ATP from the breakdown of sugars, fats, and other fuels like proteins. Now, the metabolic pathway for breaking down fats and proteins is a lot more complicated, and the main source of fuel that's used to make ATP is sugar. So glucose is our model for this. And it only works in the presence of oxygen. Everything gets backed up if oxygen is not present. Remember, catabolism is breaking things down. Think catabolism. We're breaking apart sugar for its energy. When we make energy in the presence of oxygen, we call that aerobic respiration. So if you're into aerobics, well, now you know what it's called. You're actually using that oxygen to make ATP energy to move around. Mitochondria have two membranes. They have a smooth outer membrane, and they have a highly folded inner membrane. And these little folds are called cristae. There's water everywhere here, and fluid-filled space between the two membranes is called the inner membrane space, and that's going to be important when we talk about cell respiration. And there's also the very center called the mitochondrial matrix, and that's where the enzymes are. And that's also another place where kind of important in cell respiration called the Krebs cycle takes place. And we're going to be able to compartmentalize some functions with those two membranes to make our ATP. Chloroplasts are plant organelles. They belong to a class of plant structures called plastids. Amylose is starch, so amyloplasts store starch. That's what a potato mainly is. Chromo means color, and chromoplasts store color pigments in fruits and vegetables as well as flowers. Now, we're going to find out later that there's no such thing as a science word for vegetable, but that's why they have their colors. Chloroplasts store chlorophyll and are the things that do photosynthesis. And leaves and other green structures of plants and eukaryotic algae, that's the green part, the chloroplasts. This ends part 1 of your second quarter review.